All right. Okay. Hi, everyone. It's a few minutes after. Thank you so much. Happy Friday. Thank you very much for coming to today's High Rounds. I am incredibly excited to be able to introduce my dear close friend and colleague of 15 years now. You've been here for 15 years, Jason Grebely. So Professor Grebely is the head of the Hepatitis C and Drug Use Group at the Kirby Institute at University of New South Wales in Sydney, Australia. He's actually Canadian and completed his bachelor's in biochemistry and molecular biology, and then a PhD in pharmacology at the University of British Columbia in Vancouver, Canada, and then moved over to Australia. Jason's world-leading research focuses on hepatitis C epidemiology, testing, and treatment, with an expanding focus on reducing harms that may relate to drug use, including drug treatment. He is co-editor-in-chief of the International Journal of Drug Policy, a leading policy journal in this space. He is also the president of the International Network of Health and Hepatitis in Substance Users, which is an international body for improving knowledge translation, education, and advocacy for hepatitis C among people who inject drugs. I've had the privilege of serving with him on the board for the last few years. I'm the vice president, and he'll be talking a bit about our conference at the end. Please come to Insu. But we are so pleased to have Jason here in person today to talk about, we have reached single visit testing and treatment for hepatitis C infection, now what? And before I turn it over to Jason, thank you so much for everyone that's joining online. Please feel free to kind of put your questions in the chat. We'll have a discussion for the people in the room first, and then we will absolutely get to your questions at the end. So thank you very much for joining us, and thank you, Jason.
No, thank you so much, guys. I really appreciate it. And yeah, it's amazing. I can't believe it's been 15 years we've been working together and I've never made it to San Diego. That's a bit embarrassing. And also I'm going to apologize. I just arrived last night from Sydney. So it's like 2 in the morning for me. But we'll be okay. We'll be okay. I've had my coffee. Yeah, absolute pleasure to be here. All right, here's my disclosures. All right, so before I start, some of the stuff I'll be talking about is related to some point of care testing work that we've been doing. I always like to put this up because there's so many people that have been involved in this, and it's not sort of a single person endeavor. And I just want to thank, we've got about over 150 stakeholders working together on the program, and yeah, just to acknowledge all the efforts that they've been, particularly the people on the ground, been doing a lot of work on this. So. All right, so what are we going to cover today? So it's pretty exciting. We have reached sort of single visit testing and treatment for Hep C. I think probably not surprised to this group, but I'll go through a little bit around sort of the burden of HCV among people who inject drugs and people receiving opioid agonist therapy. Excuse me, I know everybody uses different terminology, and in the States you tend to use medications for substance use disorder, but let's just go with OAT. I didn't want to change all the slides to medication for substance use disorder. And I do actually think we need to be really careful about our terminology and what are we actually talking about. So that's a whole nother talk, but let's not go down that path. A little bit about what we've learned about direct-acting antiviral therapy for hepatitis C among people who inject drugs, and the fact that testing and linkage to care are going to be really the major barrier moving forward. But it's super exciting. We've had some amazing innovations in testing, and hopefully I'll run through some of those with you. But I think one of our key challenges is how do we sort of maintain providing support for people once they get onto treatment? We're starting to see people sort of drop off, and so what can we do to help support people through their journey? And I think one thing I hope I can communicate to you is that we're not doing things at great enough scale. We've got lots of sort of clinical trials and pilot studies, but we're not delivering them at the scale that we need to actually have an impact. All right, so the burden of HCV among people who inject drugs and people receiving OAT is considerable. There's updated estimates from Homi Razavi's group, so Polaris, demonstrating that there's 57 million people living with Hep C globally. About 5.8 million people are recent people inject drugs. So the prevalence is about 39%. Louisa Degenhardt and I led some work that was published in Lancet Global Health on this systematic review, which contributed to this. I think what's really important is that 4 countries, China, United States, Russia, and Brazil, actually make up more than 50% of all the infections globally. So if we're going to have any chance at success here, we really have to make sure that we're sort of focusing in on those countries. All right, So where have we come in terms of direct-acting antiviral therapy among people who inject drugs? Look, I think it's pretty impressive. If we look at, this is a systematic review that was done by one of our postdocs, Behzad Hajarizadeh in the Lancet Gastroenterology and Hepatology. And if you look at people that are receiving opioid agonist therapy for substance use disorder, this overall sort of cure proportion is 91%, so pretty good. And if we look at people that have recently injected drugs, 87%. When we did a meta-regression analysis, what was interesting is that, not surprisingly, is that clinical trials were sort of associated with a higher response to therapy, so about twofold greater. So not surprising, you get sort of some loss in fidelity as you sort of move into the real world. But the interesting thing is that the difference was predominantly due to loss to follow-up. It's not like people were failing virologically, and so a lot of this is due to reinfection, but also loss follow-up. Talking about reinfection, I think we have to acknowledge that it's going to occur. This is a different systematic review that Behzad led, looking at reinfection following successful treatment. And again, in people on receiving opioid agonist therapy, the rate of reinfection was 3.8 cases per 100 person years, or about 3.8% per year. And then slightly higher in people with recent injecting drug use, not surprising. So 6.2 per 100 person years. I think the key thing is that we have to acknowledge that reinfection is going to occur. We need to ensure that we have systems in place to rapidly detect reinfection, retreat the reinfection without stigma and discrimination, because that's going to help prevent transmission to or ongoing transmission to other people. So I don't know. I know it might be harder in the US. In Australia, we have no restrictions on who we can treat. So it doesn't matter. We've got some people in the prison system who've been treated up to 7 times. But it has no impact. It doesn't matter, you know, in terms of our health, the way the healthcare system is sorted that out. It doesn't matter in terms of the cost being provided to the sort of at a lower level. Okay, so testing, diagnosis, and linkage to care is going to be really one of our key barriers. This is from the same review that we did with Louisa Degenhardt. Again, Behzad led an analysis looking at testing HIV and Hep C testing and treatment uptake globally. So overall, 47% of people who inject drugs have received HCV antibody testing. And you can see it ranged really widely from less than 1% to 93%. Again, treatment uptake pretty unimpressive, I guess, ranging from 2% in India to 89% in Spain. And the treatment uptake was quite low, so less than 25% in 14 countries, moderate being 25 to 75% in 8 countries, and only 1 country in the world had a treatment uptake above 75%, and that was Spain. So we really have further work to do if we're going to lift these testing and treatment numbers. So I mentioned that the burden in globally, I think predominantly this group's probably, I think, maybe more of an HIV focus group, but there's a national sort of strategy, or there's the global health sector strategy from the WHO. And the goal is to eliminate viral hepatitis as a major public health threat by 2030. I mentioned in Australia, we're now on our 6th national strategy, which is amazing. There's a number of sort of key features of the program, but having these strategies in place has really been quite important at a policy level. And just to show you, this is some data from Australia, and you know, in 2016, as we had the direct-acting antiviral therapies that were approved, we had this massive increase. I think most countries would see a similar figure, the US being similar as well. So you had this, you know, massive increase of all these people that were waiting to be treated, that got treated, and then as we sort of get to the more difficult populations, the treatments have gone down. I think, and then sort of COVID didn't help. I think what's exciting is that in 2023 is the first year that we've actually seen a rebound. And I'll talk to why that might be, I think, but we still, you know, we need some modeling to help us to sort of understand what the counterfactual would be. I'd like to think that it was some of the testing programs that we've been putting in place. And I'll explain some of that in a second. All right, so why do we have these low treatment uptake for hepatitis C? You know, I'm sure a lot of the clinicians this would resonate with. You know, it's a pretty long process. You think about it, you send somebody, they get their antibody test, and you have to go send them off to get the venipuncture done at a pathologist. Then they come back, and then you do the RNA test, and they come back. And look, sometimes you can simplify this, but it's a no-brainer that why do we have sort of this loss to follow-up? It's a lot to be asking of patients. I know how bad I am at sort of sorting out my own pathology, you know. So anyways, so I'm gonna talk through what I think are some things that we've done and I think why they've been successful. So a couple things. So many people are probably aware of dry blood spot testing. So finger stick onto a card. We can then send that off to a lab. Usually it's 5 spots. Send that off to a lab. We can do HIV, Hep C, viral sequencing, all sorts of stuff we can do on that. Point of care RNA testing, which for you guys, you should be excited. You've got access to point of care testing for Hep C. The gene expert now in the US just got FDA approval. So I'm excited for you. And we've also got some point of care antibody tests. So you've only got one option, the OraSure or a quick test. So which is fine. It's just too expensive anyways. But there's some other technologies coming through, and I think we need to be pushing them. For example, the one on the right is the INSTI. I don't know if you guys have seen that. It's a one minute point of care antibody test for hepatitis C, and they actually have a syphilis HIV combo test that's just been approved by Health Canada. So, and I've convinced them they're going to make me one that's HIV, Hep C, syphilis, all on the same test, and they promise me next year they're going to get me HIV, Hep C, Hep B surface antigen syphilis on the same test with the control dot in the middle. So, anyways, I think multiplexing is the way to go. We need integrated sort of disease testing models and stop having HIV and Hep C and everyone in their own separate rooms. We need to sort of bring this stuff together. Okay. So we did a systematic review a couple years back, again, led by one of our postdocs, Evan Cunningham, that was published in Lancet Gastroenterology and Hepatology. And so I'm just going to try and break down, it was a lot of data, so I'll try and break down for you sort of what was found in terms of interventions that are successful to enhance testing and treatment. So the first thing is this is looking at sort of, well, what are things that we can do to simplify testing? So we know that, and so this is sort of the cascade of care, if you will, antibody testing, RNA testing, linkage to care and treatment. If the box is shaded, it tells us that there was evidence that there was an effect of this intervention on that particular step of the care cascade. So we have data that sort of the things that we can do, so point of care antibody testing, point of care RNA testing, dried blood spot testing, reflex RNA testing, I'll explain that in a second, and an opt-out screening, obviously with consent. Okay, so what is the first thing? So the first thing that we should be doing is we should be providing on-site testing. It's a bit of a no-brainer. The minute that you have to refer somebody off-site to a pathologist to get blood work done, you're going to potentially lose people. So if you can do the testing on-site and turn that from 5 visits into 3 visits, that's a bit of a no-brainer. Okay, so then once we've got testing on site, what else can we do? The first thing in the US, you guys have done a great job at this. I think partly because there's some money in it for the labs, but still, I think you were on the ball pretty quickly. And so reflex RNA testing. So what that is that if you have a patient in front of you, you either get 2 samples collected, so that what can happen is that when you send those samples off to the lab, the first tube is tested for antibodies, and then if the antibodies are positive, the lab automatically tests the second tube, which is collected in the proper tube for RNA test. And then you can immediately ensure that the person gets a test for current infection. I actually don't think it's ethical or we should be actually testing people for hepatitis C unless we're testing for current infection. It makes no sense. And I know it's a cost thing, but you want to know, does the person in front of you have hepatitis C, not if they've been exposed. So I think it's great. US has done a fantastic job of this. Lots of labs are doing this as standard, cost effective, it makes sense. What's the next thing we can do? Well, we can combine that. So why don't we do a rapid point of care antibody test? Then if they're positive, then you take the venipuncture draw and send it off. And then that saves a step there. So I think that's another potential option. And you guys, the beauty as well is that you've got the CLIA waiver system and people can be trained up to do that. What about dry blood spot testing? So this is another way, obviously the problem with both of those options is that you still have to get a venous blood draw, which is a problematic issue for people who inject drugs who often have poor venous access. So dry blood spot provides us with one option that can get around this. So this is obviously this spot on a card, gets sent off to the lab. We don't have to do venipuncture. I've been really fortunate. I've been working with the Ministry of Health in our state over the past couple of years, and they've been running a project looking at dry blood spot testing implementation at population level. Initially it started off as an HIV home testing project, and then facilitated testing was added in. And Hep C in 2017 was then added in. And then in 2018, this assisted registration started. We started doing testing in the prisons then in 2018, and you can see in 2020, there was a slight dip, but people sort of pivoted and found a way around that even through sort of the COVID period. So it actually provided a great testing method for people during COVID through home testing and through also some facilitated based testing. So they tested just under 8,000 people across that period. I think we're now at, I don't know, maybe double that, maybe triple that. And the Hep C prevalence was about 15%, so a bit lower online, 5% in online, 17% in the community, and 14%. And that's current infection prevalence. That's not antibody, that's current infection. And treatment uptake, pretty impressive, 45%. Bit lower in the online group is small numbers. That being said, the one thing is that for the HIV sort of population, the self-testing worked reasonably well, but for people with hepatitis C, the self-testing or home testing option there wasn't a lot of uptake there. I think we're talking hundreds of people. And then 26% treatment uptake in the community and 63% in the prison. Just to give you an idea of that community treatment uptake, so the prison uptake, great. That we have a statewide data linkage project where everybody notified with hepatitis C, we know they're in this database. And what we can do is match that. That's linked to treatment uptake for the entire country. So that's linked into that. So just to give you an idea, the treatment uptake in 6 months in the overall population of people with hepatitis C in our state is about 26%. So that was on par. And that's in the past sort of 6 months. If you look at ever treated, it's probably closer to 60%. Anyway, so I just want to, so this performed probably about as well as standard of care in the community, but in the prisons, the treatment uptake actually with standard of care was probably is 26% in that linkage project. So it achieved quite a pretty good uptake, but 63% in the prisons. And that's, you know, you have nurses going into like the cells with people putting their hands out, doing testing, or out in the wings. So it's a great tool for that setting. All right, so but dry blood spot testing's okay, but it's not it's got lots of limitations. You still have to wait for the results to come back. You get people lost to follow up. So we really wanted to sort of figure out, well, how do we kind of get things moving? So we had sort of, this is the Cepheid Gene Expert. You guys seen, so it's PCR based system, sits on a table, about the size of a printer. This is what the cartridge looked like. And it was amazing. Is back in sort of 2015, I got invited to this dinner with this company, and I was like, oh, what am I doing? You know, I kind of knew about them, and it just so happened. So this is Dave Persing, and he's the chief medical officer, and the guy was sitting next to me. So I had a chief medical officer, I didn't even know him, sitting next to me. We got chatting during dinner, and they're trying to sell me on this machine. So it was 2 hours, and it required venipuncture, and I said to him, no way, forget it. You guys are crazy. I said, your population that you need are people inject drugs. There's no way people are going to sit around for 2 hours. He says, wow, I think we can get it shorter. I said, okay, well, it doesn't matter. You're still going to have an issue. You're going to take venous samples. And he said, well, we've been working on a finger stick version. I said, okay, that's exciting. I said, well, we've got this cohort that we've been running, and where we've been like recruiting sort of people over sort of a 1 week period at a drug treatment clinic. And we do sort of a sample of that over a week, can recruit like 100 people, you know, over that week period to look at, we were looking at Hep C prevalence and linkage to treatment. I said, why don't we can we evaluate this test before it goes to market? And he said, well, we don't have a test. I said, doesn't matter. Let's just give it a go. So what we did, so this is a finger stick into what's called a mini vet. Now, be careful, because this isn't the version that you guys have in the US. The one in the US, you have to, I'll explain it in a second. But so this is a mini vet, 100 microliters, EDTA coated, and you essentially finger stick into this tube. Got that little orange thing as a plunger, and you put it inside the top of the cartridge and push it down. And then you put it inside the machine, and that's actually Francois, one of our, he now actually works for Cepheid, which is funny, but anyways, he was a lab scientist in our group. And this is one of the syringe service programs up in Northern Australia where we had 4 of the gene experts in parallel when we were doing this validation study. And we gave it a go, and it was the first proof of concept of a finger stick point of care RNA test. We used the venous cartridge, so it actually took 2 hours to run, but we actually just, and you had to have 1 ml of blood, so we just put it up to 1 ml of buffer and gave it a go, and it was fine. I think it was 99% sensitive and specific, so it fared really well. And then Cepheid used that data to develop a finger stick version. We evaluated that 1 hour, 100% sensitivity and specificity, actually 58 minutes. And this really has been a game changer because it allows us to do single visit testing and treatment for hepatitis C. People find it much more acceptable to venipuncture, and we've done some work on that, and it does reduce follow-up. So we did a couple of pilot studies. The first one we did in prisons called the Pivot Study. And what we did was we had a period of standard of care, and then we rolled in the intervention, so there's sort of one-stop shop. And we demonstrated that we could improve the time to treatment from 99 days down to 6 days. And then we increased treatment uptake from 22% up to 93%. So pretty remarkable. And we also then piloted this sort of peer-led sort of program at one of the syringe service programs and showed that we could get an 81% treatment uptake. So pretty cool. Oh yeah, so the version that you guys have is a bit different. It's a finger stick into sort of a BD microtainer, a microvet cartridge, which you'd use for infant collection anyway. So you get it in, put it in there, then use a pipette to take it out and then put it in. Problem is that the mini-vet tubes aren't FDA approved, so they couldn't use that. So these ones are, and it'll work. You'll be fine. So really exciting, really cool. So I just starting to think of things more of in an impact framework. So we had these sort of trials and these projects that led to these molecular evaluation studies, and Cepheid used, so who used this information? Well, Cepheid used this to develop their new test. The EU and the Therapeutics Goods Administration and now the FDA actually have used the data to approve the test. So it's CE marked first, and then it was TGA approved and now FDA approved. It's now, point of care testing is now integrated in all the global sort of guidelines from WHO and EASL and hopefully AASLD. They're soon, I think they're updating it now. And what's really also important is that they've been embedded in the national and state and territory strategies. Yeah, sure.
How do you get people to stay 58 minutes?
Look, not everyone does. It's interesting, in the syringe service program, 80% of them stayed that day, and they usually come back, so they get a text message and say, oh, your results back. And then the other ones came back in the next couple of days. Look, for that one, there was a financial incentive of $30. Look, it's tough, and that's why I'll go through some other things here about why I think we need to modify our approach. So the cool thing is that we can use point of care RNA testing for initial diagnosis. We can confirm cure, and then we can also use it post-treatment for reinfection monitoring. Okay, so I do want to caveat this, and it took me several years to convince all my Australian colleagues and the nurses and the doctors to go this way. So I'm trying very hard to push you guys in the US not to make the same mistake, okay? So everyone wants to use the machine right up front, for everyone. They just want to go and point of care RNA test everyone because it's a fancy new machine and they think it's going to be the easiest thing. It is not, all right? These machines are quite complex. It's really hard to train up the operators. They get frustrated using them. You forget how to use it. You put it away for a couple of weeks. You come back to it. You forget how to use it. So yes, it's really exciting, fancy, shiny, new sort of machine, but it's not, there's only one setting in all of Australia that I would say we should be using immediate point of care RNA testing, and that's in the supervised injecting centers, because the prevalence of hepatitis C is like 80–90%. That is the only place where I think it makes sense. In all other settings, I think we should be doing point of care antibody testing with then immediate reflex RNA testing in the people that test antibody positive, okay? And there's a couple reasons for that. First of all, the point of care RNA test is way more expensive. Look, I don't know what you guys are going to pay. This is Australian dollars, so it's like $4 US. It's $10 for the antibody test and $60 for the RNA test, okay? The other thing is point of care antibody testing is it's a lateral flow. It's so much easier to train up peers, train up whoever to be doing that. Look, we've trained peers up in the point of care RNA platforms. Don't get me wrong. This is not rocket science, and you can train anybody to do it. It's great. We've got a huge peer workforce. Some of our best operators are peers. So I think that's really important. I mean, we've really tried to push that. But it's much easier to train operators. Just to give you an example, our point of care antibody training is 1 hour. Our point of care RNA training is about 7. So, you tell me which one the nurses are going to want to do. So the other thing is that there's much higher improved acceptability of point of care. Oh, I'm going too slow. Okay, I better speed up. There's improved acceptability of point of care antibody testing among patients and providers. It's about, like I said, 58 minutes as compared to the antibody test, which is much shorter. The other thing is if you're doing high intensity testing campaigns, so where you go out and, test a bunch of people, you're limited to 4 tests per hour on that machine, right? 1 test per hour per module, right? If you go and use the antibody testing first, let's just say that you're out, and you screen 20 people, right? And the prevalence is 20%. Of those 20 people, 4 will be positive. So you can immediately increase your throughput from 4 tests an hour to 20 tests per hour. And this is really important. Like we go into the prisons, and I'll talk a little bit about it. We test, we'll test 150 people a day. And so the key rate limiting factor is actually the gene expert machine. So we use 5, 6 in parallel when we're out at the prisons. They're like, they're not cheap. They're 64,000 a machine. And the other thing is the cost effectiveness. So what we did is we did some Sophie Shi from our group did some work around this. And let's just take the prison. So I told you the standard of care was about 60% ever. So we use that as kind of a conservative estimate. And then here's the treatment uptake, and let's just take the prison. So 93% from that study that I showed you. So the cost per person treated with standard of care is about $1,426 per person treated. You get a small benefit with if you were to point of care RNA test everyone. But if you use the point of care antibody testing and the reflex RNA testing, you actually save about 1/3. And this is because of unnecessary point of care RNA tests in the people who are antibody negative. So you're saving you're saving those unnecessary tests. And actually, if somebody's received treatment, it makes 0 sense to antibody test them. So you don't do an antibody test in those people. So you get some further benefit if you just say, well, if you receive treatment, you'll go straight to an RNA test and not receive antibody testing. And this is a complex slide, but I'll just kind of run you through it. So here is average cost per treatment initiation on the y-axis, and on the x-axis is the antibody prevalence. So if we start at the far left, for those people online that can't see me pointing, and let's just say the antibody prevalence is 10%, okay? The most expensive thing to do is in blue, and that would be point of care RNA testing for everyone, then standard of care. But you can see that the 2 point of care antibody and reflex RNA testing options are the cheapest. And it isn't actually until you hit an antibody prevalence of 35% that the immediate point of care RNA testing for everyone becomes more cost effective than standard of care. And actually, it's not until you hit nearly an 80% antibody prevalence that testing everyone with this immediate RNA testing approach compared to the 2 antibody testing strategies becomes more cost effective. So we should always be using point of care antibody testing and then reflex RNA. Or if you think that somebody's had a really recent exposure and you think that there might be, that they might not have seroconverted yet, then go ahead and do an RNA test in them for sure. Because some people won't have seroconverted yet and you want to identify that RNA early because that's what appears first. Yeah.
Where does genotype testing fit in? I'm sorry.
No problem. Genotype testing, we can throw that in the garbage now.
But what is this?
I started some controversy.
Yes,
It's great. I
definitely.
love it. I
And
love
then
it.
we can talk about that.
Okay, let's talk about it at the end. Let's talk about genotyping. Alberta, for example, they said, we are not going to do any more genotyping. We're going to invest it all into reflex RNA testing. And you'll tell me, but how are you going to distinguish reinfection? And then I'll say, well, you've got 2 regimens. What were they treated with first? And unless they have compensated disease, what's that, sorry?
Did you say so?
I said I'm lazy.
I'm lazy. But it's very expensive. It's an expensive test. And for example, anyway, let's go there later.
Yeah.
I realize I'm going too slow, so we can have this discussion, but good discussion. Okay, we're really fortunate. The stuff I'm going to tell you, we were so fortunate that there was a program in place with the Kirby who already had a system set up for point of care testing using the gene expert for STIs and COVID. Just to say that Rebecca Guy and Sue Matthews, and if they hadn't have had their program set up, there's no way that we could have hit the ground running as quickly as we have with our program. So we were just important to acknowledge people for the work that they've done. And this is their remote network that they set up for COVID, remote rural network that was funded to do. And now it's sort of respiratory testing for flu A, flu B, and RSV. So we got some funding from the federal government to set up a point of care program. So it's the Australian Hep C point of care testing program. The plan was to test 60 or set up 65 sites nationally, test 50 to 60,000 people. Pretty broad drug treatment clinics, syringe service programs, prisons, mental health, mobile outreach models, homelessness services, and Aboriginal community-controlled health organizations. Pretty broad testing inclusion criteria. Pretty much anyone at risk of Hep C, or if they attend a service that provides testing. all the program includes everything, standard operating procedures, logistics, deployment, setup of the Gene Expert platforms. The trainings delivered by Flinders University at the International Center for Point of Care Testing. We developed a quality assurance program with the National Reference Lab. So this is twice yearly blinded competency or panels, quality assurance panels, 1 negative, 4 positives, genotype 1 low, genotype 1 mid, genotype 3 low, genotype 3 mid. And then monthly competency panel testing, which includes unblinded positive and negative samples. So pretty rigorous. We've got an IT connectivity system, so the results go directly off the Gene Expert laptop into through a sort of a glorified email encryption server, which then comes in identified into our database. We can also send it identified back to the health departments, and it can go back into the clinic records. And we have sort of a research and evaluation framework that we've set up. We've got sort of NHMRC and MRFF funding of somewhere around sort of $12 million to sort of evaluate the program. Okay, so here's where we're at right now. So we've got 102 sites set up. You can see that predominantly in sort of the urban areas, but we do have almost a machine in every public health network in the country. But that's a bit deceiving because they're actually doing testing at lots of different places. So it's not one site. So we've actually tested in 391 testing locations now across the country. We've got 36 prisons that are actually, we've set up Gene Expert platforms or delivered high intensity testing campaigns. This is a bit old. I think we're up to 26 high intensity testing campaigns now across the country. We've trained 360 operators, done over 32,000 tests, and have, I think this is again just a touch old. We've got over 3,500 people now with current Hep C that have been identified. I took an older snapshot of data because the only thing is that people come in at the 12-week post testing time point. So we're just cleaning some of the data. But so this is treatment uptake based on data that I know is clean. But a third of people are for retreatment, have been coming forward. But treatment uptake overall is 77%, 50% in the community. Again, we're working through the cleaning. We actually think that the data is probably closer to 65% in the community. But what is so striking is the prison. I told you 93% in a kind of well-controlled clinical trial, 91% treatment uptake in the prison in real world. So that's just amazing fidelity. It's just so impressive. So we're doing about 10% of all the treatment in Australia and about 25% of the treatment in Queensland. Okay, I need to move. Okay, so multidisciplinary approach has been really important. This has included sort of the diagnostic evaluation, the pilot sort of trials, the cost effectiveness, some qualitative work looking at barriers and enablers to implementing testing strategies. We're just starting to do a lot of work around what works and why. So implementation science to identify the barriers and enablers and specifically design strategies to address those barriers and enablers and then test those in larger cluster randomized trials. And I think the key question is going to be how do we further drive policy change and some of the investment case work on that. Okay, just a couple of reflections on success factors. You know, there's been a lot of luck. I'll tell the story really quickly. I actually emailed about a week before the Australian Viral Hepatitis Conference in 2023. I emailed the health department, said we want to set up a program similar to this STI and COVID program. Can we get $1.5 million to set up 20 sites? And we invited the health department head to the conference, and he was there, and he's like, oh my God, saw all this data on point of care testing being presented. He phoned me up a week later and said, Jason, what could you do? Pick up $500,000. And I'm like, a lot more. And so put the proposal together. He went, and we were lucky that we had a really supportive advocate. The policy window opened, and he went around and found end of year's financial underspend to like pull it all together for us. So a lot of luck. Things kind of snowballed. This started off a $70,000 seed grant from the university and just partners coming in, and it just kind of avalanched as it went. We've got an amazing operational team. It's really interesting. We're running this program as it's a normal program but it's all embedded within a research framework. Everyone's consented. All the sites are through ethics. I think we have like 20 ethics committees sort of. So it's the team is amazing. We've got an amazing operational team. Healthcare champions, the nurses, doctors on the ground. We've got a couple that whenever there's an issue, Mim, can you please come? And you're going to see Mim later. We will get to the video. Whenever there's an issue, that she'll come and have a chat on our community of practice and be like, okay, guys, come on, this isn't so tough, and get everybody on board. Relationships matter. I meet with every single site. I know all the people at the sites. I think that matters a lot. And when you need a favor, again, the golden rule, do unto others is how you'd want them to do unto you. So always trying to do stuff for sites so that when you do need a favor to be involved in XYZ sub-study, they're always kind of willing to be. And I think measuring the effect of having this research framework is really important to be able to sort of evaluate the impact of this and the success. Okay, so what we're doing now is that if somebody comes through, we ask them, have you ever been told you have Hep C? Because if they yes, even as an antibody, it's usually pretty good in terms of their awareness. So if not, then they get an antibody test. This could be a lab-based test, it could be standard of care, or it could be a lab reflex test, or they can have a point-of-care antibody test. And if it's positive, they have a lab-based RNA, a dry blood spot, or a point-of-care RNA. So you have a lot of different options. If yes, they have been told they have Hep C, go straight to an RNA test, and that could be a lab test, a DBS, or a point-of-care. And it may be surprising, but we were slow to the mark on having an antibody test, and we just had our first antibody test approved by our FDA equivalent in May. So that's made things a little bit easier. All right, so it's really critical what's the right test for the right patients in the right setting. I've talked a lot about point of care, but there's a lot of settings where point of care testing is not going to work. If your prevalence is really low, you're going to get no benefit of the Gene Expert platform. So we have prisons now where their prevalence is down to 1% current Hep C prevalence. Training up all these people to use these Gene Expert is pointless. So they've implemented a model with us where we're doing point of care antibody testing and then just reflex venipuncture testing. Then we can do the HIV, the syphilis, the chlamydia, the gono, all the other stuff that you need to do that the Gene Expert is going to help you with. So the prevalence of infection is important, cost effectiveness of testing in that setting, timeliness of how quickly you deliver the results, whether you can link them to care. Somebody that's coming every day may not need a Gene Expert. Capability to perform the point of care testing and the point of care testing counseling, it's a lot of work. And then access to the platform and the program, it's expensive, and having the available resources to deliver it. And whether or not you're mobile. So a lot of these are kind of going out in mobile models of care. All right, so this is the last kind of couple of bits. So in terms of, we talked about simplification testing. Well, what about patient engagement in care? Some really nice models this is one from New York City, looking at patient care coordination. So what they do is they assign people to a care coordinator that supports them navigating the health system, accompanying to medical appointments, medication adherence. You guys are probably doing a lot of this stuff for HIV as well, and there's data from HIV. So they matched patient care coordination model to match a cohort of people with HCV in the New York City Surveillance Registry. So in the patient coordination arm, 72% initiated treatment compared to 36% in the matched cohort, and 65% had cure compared to 47%. So patient care coordination, really important model. This is some data that was just presented a couple months ago by Sean Faladnawulia out at Hopkins, looking at peer support in drug treatment programs called the RAPID study. And what they did, and what's nice is we're starting to see some randomized controlled trials. We haven't had this, and that's why I wanted to include this. But in the Hep C field, HIV, you guys are a little bit more advanced in some of the randomized trials. So what they did was randomize people to on-site treatment with peer support versus referral to a local specialist. So the on-site treatment with peer support, 89% initiated treatment as compared to only 21% of those referred to the local specialist. Median time to treatment was 19 days compared to 50 days, and 61% achieved cure as compared to 14%. The only issue is that the two arms were very different. One is looking at on-site plus peer support versus referral off-site. So it doesn't really tell you what the effect of the peer support component of the model was, right? But we've got some data from the UK. They've actually done lots of work on peer support. So this is a retrospective analysis of the English National Health Service Treatment Registry, over 30,000 people between 2017 to 2020. So what they did was they compared patients managed with peers versus patients not managed with peers. And this is really small. I don't expect you guys at the back to see it, but just trust me. Treatment uptake was higher in the peer support group, about 12% higher relative risk. And the proportion completing treatment of all expected increased, and the odds ratio was 2.45, so about twofold greater with the peer support. So, but we need more randomized data, I think. This is helpful, but this is sort of observational. This is a really cool study. I put this in, your colleagues up in San Francisco done this. I'm not sure if you've seen this, but it's called the No One Waits study, so the NOW study. It was a non-randomized, single-arm, open-label trial, and what they did was they screened with point-of-care antibody tests and then did venipuncture with a 1-week turnaround, and then people returned 1 week later. And if they were positive, they were given a 14-day starter pack of sofosbuvir valpatisvir, And what they did is they took the dose on that enrollment, and then they had 2 weekly visits. And the overall proportion initiating treatment of the 89 that returned for their RNA results is 98% and 67% had cure. But the SVR in those that completed treatment was 84%. So I think this is we've got people to this stage, we've shown that we can increase treatment, but what we need is all of these additional things: peer support, patient navigation, starter packs to get around the pharmacy issues, telemedicine, I didn't show that, but remote prescribing to get over that barrier. And so these things are needed. Provider engagement in care. I just want to say, let's not forget the stuff that's simple-ish. Medical chart reminders. This was the single most effective intervention in the systematic review. So for increasing testing, medical chart reminders, either physical notes in the thing with a little like sticky flag, or actually a more sophisticated like electronic system, 7-fold greater testing uptake and 4-fold greater linkage to care. So, the first thing we should be doing is let's do a medical chart audit to figure out what we're starting with if we haven't had that in place. We do some provider education at INSU, got a big toolkit, go on the website, have a look, but provider education is obviously really important. And then just lastly, if you do a bunch of stuff, it's going to work more than doing one thing alone. So things that had multiple components to it did the best for both testing and linkage to care. So what is the future? So if we think about strategies to enhance testing, look, I'm so excited for you. I know you can tell I'm a pretty excited person, but for you guys to be able to use the point of care testing in the US, it's really going to help you kind of move things forward, but it's not a silver bullet. It's also a massive pain in the ass. And you really are going to have the systems set up to be able to deliver the point of care testing. It's a lot of work. So you just have to acknowledge that, but then go in eyes wide open and just acknowledge it's going to be a lot of work, but it will help. And I think what's really critical is that point of care testing is not going to be the best fix for everything. So we really need to figure out what's the right test for the right patient in the right setting. And let's not forget about the simple-ish things, because I know sometimes setting up a medical record audit or electronic systems is not the simplest. And we've got red flag fatigue by clinicians of all the stuff that pops up that they need to be doing. So I get that. This is one that I think we need to think more about, but can we use geo-mapping data to better target our testing? And should we be integrating HIV and Hep C and syphilis and trying to be able to better use that data in a more real-time way with the health departments to target where we haven't been doing testing? In terms of strategies to enhance treatment uptake, I think we've got lots of innovations to overcome these barriers, and we're working on these starter packs, telemedicine, financial incentives. We're going to go one step beyond the starter pack idea. We're just going to provide the whole treatment up front, because I just think, look, we're lucky. I don't know if your health system will allow you to do that, but ours will, so we'll do that. But I think the one thing we really need to kind of work on is these interventions to support people through treatment. Yes, we're getting people on treatment, but if they don't cure, it's all for nothing. Well, it's not all for nothing, but it's close. And how do we integrate peer support and navigators into practice? How can we co-design solutions with the community to amplify the enablers and address the barriers? I think some of this implementation science is really important. And what about post-treatment follow-up for liver cancer and other things? And I think this really needs increased funding to support the infrastructure to do this. We need funding for nurses, funding for peers, patient navigators. Everybody's expecting us, well, the nurses and doctors, to do more with the same amount or less, and it just can't, it's not sustainable. And I think we're really going to need to, if we're truly serious about elimination, we have to move beyond these pilots and get the governments to fund things and deliver these interventions at scale, because we can't have pharma, they're not going to fund the health system. They fund these pilots to generate the data, and we need the governments to kick in. And I think we need innovative models maybe of how we do that. And I think just lastly, I didn't talk a lot about this, but I think at the core of this is stigma, discrimination, criminalization, and violence among people who inject drugs. And until we sort out some of that stuff in terms of health-seeking behavior and people fear of attending hospitals and all that sort of stuff, we're going to be, really going uphill. So again, massive thanks for all your time. Again, thanks to everybody who's been involved. And I just have one video to leave with. I think we're still okay for time. We'll have about 10 minutes for questions. So I'll leave you this. What I think this is nice, showing you how things kind of can work on the ground in just a really fantastic group that's been doing work in Australia. Oh, before we get there, I have to talk about INSA really quickly. So conference is next year. This is International Network on Health and Hepatitis and Substance Users. Natasha is vice president, and it's a great group. It's a good family. So if you are interested in this sort of stuff, come to Cape Town. If you're an early mid-career researcher or a PhD student, it's free to become a member. So become a member and be engaged. And Cape Town's a beautiful place. So October. So I'll leave you with that, and we'll go to the video. There we go. Do I need to push? There we go. Point-of-care testing for hepatitis C is quick, painless, and easy, and people can essentially get tested and treated in a single visit. It's been a major game changer in terms of management of hepatitis C infection. That's my fault. Compared to traditional laboratory-based testing, point-of-care testing has been shown to increase linkage to care and treatment.
The National Australian Hepatitis C point-of-care testing program actually looks to scale up point-of-care testing and linkage to treatment in a range of settings, including mobile and community outreach, OST services, corrective services.
Tony was what got me over the line. I probably received the hepatitis C treatment because of Tony. He was what got me over the line.
With Gavin, I remember this moment. I was saying, possibly for the third time, that we can have your results and have your medication in your hand today. There was a moment where he just kind of acknowledged that, you know, by the end of the day, his life could change, and he took it.
The ability to have peer workers leading the test and treat model is very unique and also provides lived experience on which to draw from and make the whole program relevant to its end users.
So the van is set up with a needle exchange at the front. So we have a disposal bin so that anybody can pull their used ones into the disposal bin.
Jodie still keeps in touch with me. She is great. She's helped a lot of my friends, and a lot of my friends are not nervous, embarrassed to go back to her. Simple as that. And she does not judge anything or anybody at all.
Some people don't have the best veins to do full blood tests, so the point of care with just a finger prick is amazing. It takes less than 5 minutes to do a finger prick. It processes in an hour, and we have the results in an hour.
In this program, we're contributing to developing, delivering and evaluating the point-of-care operator training and quality management within the hepatitis C point-of-care testing to ensure that we have accurate and safe diagnostic methods.
Prisons are crucial for hepatitis C elimination efforts because they are a big chunk of the affected population, both in terms of carrying the burden of disease, but also ongoing transmission of the infection.
What's made the high-intensity testing and treating programs such an effective solution has actually been the advent of point-of-care testing. So what point-of-care testing is really is somewhat as a disruptor to the traditional way with which we actually have dealt with bloodborne viruses.
To date in the program, we've partnered with over 80 services to deliver point-of-care operator training to over 330 staff.
Without our more than 150 stakeholders across government, healthcare providers, researchers, and industry, we could have never achieved what we've achieved over the past several years. In particular, the work by the affected community has been absolutely essential in advocacy for the delivery of point-of-care testing nationally.
So the National Testing Program has really highlighted to us the importance of being part of a wider group we learn so much from them.
Being able to work in those dynamics with people who inject drugs and create change right there on the spot, you know, and you see it go through people's lives. It can touch, you know, so many other areas in a person's life. It leaves a space for a lot of other stuff to grow.
So post-COVID and since implementation of the national program, New South Wales data shows that we are actually back on track to reach the elimination targets, not only by 2030, but by 2028.
In terms of the long-term impact of the National Hepatitis C point-of-care testing program, the increases in treatment that we're seeing should reduce the number of new infections, reduce the number of liver-related complications, and it will help us to achieve the World Health Organization elimination goals of eliminating hepatitis C as a major public health threat by 2030.
All right, cool. Thank you. Sorry, it was the wrong film. I sent the wrong one to Marvin, but I think it works. If I wouldn't have said that, you probably wouldn't have known that it was the wrong film. But I'll get Marvin to send around. There's a really great film from the Combi Clinic. Nurse Mim you saw her, of their model of care, and it was meant to be about their model of care. And it's just really kind of impressive what they've been doing. So anyway, it wasn't meant to be that. That was our program video.
So thank you very much, Jason. Always awesome. I know, gosh, we now have like 45 people online. So great. So let's have a chat here for a few minutes and then we'll open it up to the people online. Any questions?
Just curious what your population is like in terms of mobility. You know, here in San Diego, there's sweeps and people getting moved all the time, you know, from our various areas of concentration of unhoused individuals and a lot of coming and going. So you're using a lot of injection sites. I'm just wondering about the mobility and repeat business that people have.
Yeah, there's a lot. I would say there's less moving around people move around a lot, but I don't think we have the same in terms of sweeping and people being moved from places to places. The one thing I'll say is like the inner city of Sydney. I was always so surprised when I came from Vancouver, where there's a very concentrated inner city, to Sydney, where there's kind of like there's like a drug area market, but it's not so in your face, and there's less homelessness and less, there is homelessness, but it's kind of under bridges and outside of the public view, if you will. Yeah. And so there's less, and I think that it's more dispersed. I think, and services have been developed in a way to deal with that disbursement, but there wasn't, you know, what's happened with a lot of places is with that, you know, that high value property, gentrification of those neighborhoods, and then that gets pushed to the outer suburbs where there's no services, right? That really hasn't happened in Sydney. And perhaps it's because that Sydney's had really, I mean, I'd say Australia in general has had really strong harm reduction foundation since the late 80s when the HIV epidemic came in. So primary healthcare facilities, syringe service programs, you know, and drug treatment programs that are just really well funded and well established. Does that help? But lots of mobility, lots of people moving around, lots of duplicate testing happening. Yeah, for sure.
Great.
Hey, I came in late, but I was online. It's about 8:15, so I may have missed this, but long-acting therapies are, you know, as you know, are like huge and continuing development in the HIV field. Seems to me a natural connection with what you're doing to not only diagnose people same day at the same time, but shot. You know, the antiretroviral therapies last 2 months, right?
Yeah.
And that's the treatment duration, if I remember correctly, perhaps the oral therapy is 8 weeks, right?
Yeah.
So again, I apologize if you.
No, I didn't cover it. I think it's a really good point. You know, that's kind of like, that would be the next piece of the puzzle that would really help sort things out. Can you imagine? Single visit, test and treat, here's your shot, bang. Like, amazing. So they're working on it. The University of Liverpool has been working on sort of formulations. The issue is that so glucocaprosphere, remdesivir, the sort of therapeutic dose is a slightly higher concentration than we would like for a long acting. I don't know the physical size, but I've looked. I think it's golf ball-esque. So I think they're working on getting a formulation, but it would be quite a large pellet. but it's doable. I think they've got some really fancy thing that like spiders out or something. And anyways, so yes, and it's on the plan. Yeah.
We have a pharmacist whose hand is up. I'm
Oh.
sure he can speak to it.
Great. Okay. But yeah, I think it would be a winner.
Yeah.
Lucas.
With that, should we go to Lucas?
I hope so.
Are you calling on me, Jill? Is that...
Yeah.
Okay. Well, I actually had a different comment,
That's fine.
unrelated. Yeah, thanks for the great talk. I think from the perspective of the United States, if we're thinking about elimination, the biggest barrier for us is someone having insurance to pay for these diagnostics. Because what we're running into is people, we have this great opt-out testing program in the ED, and we're identifying people who have hepatitis C, but they're underinsured or they don't have insurance, and we're figuring out ways to bring them in and basically treat them for free through our clinic. And that's not something that's widely available. So, I think if we're thinking about elimination in the US, I really believe we have to have a program similar to the Ryan White 8-HAP program for HIV, that people without insurance can be enrolled on site in a program that pays for all of this, including the treatment to then, get them treated regardless of their insurance status.
Yeah, it's such a good point, and I feel so bad you have to deal with that. And I didn't show it. I should have maybe thrown this slide in, but Natasha has done some really nice modeling that she can share with you guys in a future presentation, or has already, I don't know. But I think she's a couple things. So first of all, she's sort of, her and her group have shown that that the opioid sort of epidemic, that we've had an increase in the number of people injecting, that may tail down over the next couple of years. But when she's done the modeling, and she could probably speak to this better, she'll tell me if I've got this wrong, but when I was looking at the data, you would essentially have to treat all of the diagnosed individuals to truly get at elimination. And not only that, you'd actually have to increase testing as well, to test at least one person per year, including treating all of the diagnosed individuals. So just to your point, Lucas, if we don't sort out that funding issue, there's no way that we'll ever get there. Sorry to go to a negative point. I'm a fairly optimistic person, but I just think it's really important. And so I think, how do we do that? It's going to require a really concentrated effort. So let's just get real about it, I think, and not skirt around it. Yeah.
Okay, we have time for one more question. So Richard.
Thanks for letting me sneak in my question. You might have said this in the beginning, and if I missed it, I apologize. Did you see any differences in adherence to treatment between people who actually had to go through several steps to get to treatment versus somebody who does point of care and starting immediately? And then the other question is, if you're giving people the entire treatment at once, how do you monitor follow-up, or how do you monitor adherence?
Yeah, hey Richard, nice to see you.
Yeah.
Fantastic questions. I actually don't have data around the difference in adherence between people who immediately start versus those that have had more time to get there. My hypothesis would be is that you would get lower retention on treatment in the people that you immediately start. That being said, some of these people have had trust rapport built up at these services for quite some time, and it's kind of that thing that they just haven't got around to. So if you have that trust and rapport, maybe that'll help. So I think that the one thing that I do think is important, I think the key thing is keeping them on treatment. We know from Simplify, and also Alan Litwin shown from Hero, these medications are incredibly forgiving. Particularly the study, we haven't done as much work with adherence monitoring with glecaprevir and preventavir, but we've done a lot with sofosbuvir and valpatisvir. And if you look at the adherence map from Simplify, it's like a checkerboard, and I think what Alan's nicely shown recently in some of his recent papers is that sporadic adherence throughout is not a problem. Not that we want to, support that, but what is really problematic is big gaps of therapy at the beginning, or or in the middle. At the end, I think we're actually over-treating. So I actually don't think the end is that big of a deal if they come off a bit early. So anyways, that's my personal opinion, anecdote. Well, the data is there from Alan's study about the missing of those big gaps. But so I think we need more to kind of investigate that. Just to tell you, we're doing a trial where because we wanted to push the envelope we we've been giving people $200 to initiate treatment, $200 to come back at completion, $200 to come back for cure. Treatment uptake, we've done 36 people so far. Australian dollars, exactly. It's like $5 US. But really interesting, 100% treatment uptake. The proportion coming back to treatment completion, not so good, 55 to 60%. I talked to the nurses and I was like, what do you think, ma'am? What do we got to do? She's like, Jason, no amount of money would have helped me to get these people back. They've gone to prison. We've lost them, we've lost a phone. So that's why I tried to harp so much on that, like other stuff we need to do to support people through follow-up, because it's not just money. That's not going to just solve our problem, I guess. So I think your points are really well taken, Richard, and we need to sort it out. And just on the adherence monitoring, we do have regular contact with them. They don't just get the pills and then never come back.
Okay.
The idea is that they still have some ongoing, and we are talking about ways of doing that. And Alan's shown that you can the self-report is okay if you can play to blister pack monitoring, but not ideal. And you guys know this. You guys are all HIV researchers. You guys have been doing this forever. So yeah.
Okay, I know we're a few minutes over, so
Sorry.
thank you again. No, thank you again, Jason. Thanks so much for coming here. You're always phenomenal. Thanks everyone who's joined us online, and everyone, especially who's joined us in person. It's really nice to see everyone. Have a great week.
Thanks
Thank you.
everyone. Appreciate
Thanks.
it.